Glycolysis is the metabolic process in which one molecule of glucose is broken down to produce energy for the cell. The term glycolysis comes from two Greek words, glyco, meaning sugar, and lysis, meaning breaking. So glycolysis literally means the splitting of sugar. In this process, glucose, a 6-carbon molecule, is broken down step by step to release energy in the form of ATP, which is essential for all cellular activities. Glycolysis is an anaerobic process, meaning it does not require oxygen. It takes place in the cytoplasm of the cell and is present in all living organisms, including plants, animals, and microorganisms. Because of this, glycolysis is considered one of the most ancient and fundamental metabolic pathways in biology. During glycolysis, one 6-carbon glucose molecule is converted into two 3-carbon molecules called pyruvate. This conversion releases energy that the cell can use immediately. Now let us understand the steps of glycolysis. First, glucose is converted into glucose 6-phosphate by the enzyme hexokinase, using one molecule of ATP. In the liver and pancreas, this step is carried out by glucokinase. Next, glucose 6-phosphate is converted into fructose 6-phosphate by phosphohexose isomerase. Then fructose 6-phosphate is converted into fructose 1,6-bisphosphate by phosphofructokinase, again using one ATP. This is the most important regulatory step of glycolysis. Fructose 1,6-bisphosphate is then split into two 3-carbon molecules by the enzyme aldolase. These molecules are dihydroxyacetone phosphate, or DHAP, and glyceraldehyde 3-phosphate, or G3P. DHAP is quickly converted into G3P. So now we have two G3P molecules. Next begins the energy-producing phase. Each G3P molecule is converted into one 3-bisphosphoglycerate by the enzyme glyceraldehyde 3-phosphate dehydrogenase. During this step, NAD+ is reduced to NADH. Then one 3-bisphosphoglycerate is converted into 3-phosphoglycerate by phosphoglycerate kinase, producing one ATP per molecule. Since this happens twice, two ATP molecules are formed by substrate-level phosphorylation. In the final steps, 3-phosphoglycerate is converted into 2-phosphoglycerate, then into phosphoenolpyruvate, or PEP. Finally, PEP is converted into pyruvate by pyruvate kinase, producing another ATP per molecule. This step also occurs twice, generating two more ATP molecules. Now let us summarize glycolysis. 2 ATP molecules are used in the investment phase. 4 ATP molecules are produced in the payoff phase. So the net gain is 2 ATP molecules. Along with ATP, 2 NADH molecules. Water and 2 pyruvate molecules are formed. So the final products of glycolysis are 2 pyruvate, 2 ATP, net gain, 2 NADH, and water. This completes the glycolysis pathway.